Welcome students. Rayat Shikshan Samsthan's project Rayat Quality Improvement Program, which was run to guide students about MST CET, NEET and JEE. I am Sir Sujata Dheeraj Patil from Rayat Shikshan Samstha's Dahivadi College, Dahivadi, Taluka Man, District Satara. We actually study about the topic photosynthesis from 11th standard from biology subject. Let us study about the photosynthesis. In this topic, we actually study about the mechanism of photosynthesis. Firstly, ultrastructure of chloroplast. What is chloroplast? Chloroplast, it is the cell organelle in which actually process of photosynthesis it was carried out. Then we also study about the chlorophyll and different types of accessory pigments. We also study about the nature of light and the quality of light. And at the end, this topic we study about the mechanism of photosynthesis. Then we study ultrastructure of chloroplast. What is chloroplast? Chloroplast, it is the cell organelle in which actually process of photosynthesis it was carried out. Then we study about the chlorophyll pigment and the accessory pigments which are involved in the process of photosynthesis. We also discuss about the light, that is quality of light. And at the end we discuss about the mechanism of photosynthesis in which two processes as light reaction and dark reaction was done. Light reaction, it is the process during which ATP's energy is synthesized which was required to carry out the process of photosynthesis, and dark reaction is the reaction or the process during which actually CO2 was reduced to form carbohydrates or food synthesis of. What is photosynthesis? Photo means light, synthesis means to prepare. Photosynthesis is the process during which light energy it was converted into chemical energy, or it is the process in which O2 is evolved. Where this photosynthesis it was carried out? This photosynthesis it was carried out in the cell organ which is called as a leaf. Then what are the raw material which was required to carry this process? The raw material which was required to carry this process of photosynthesis, it was CO2, carbon dioxide, water, chlorophyll and light. How we define photosynthesis? Photosynthesis it is defined as it is the anabolic and intracellular process in which there is a chemical reaction between carbon dioxide and water in presence of light. With the help of chlorophyll, food is synthesized and O2 is released. As we know, plants are autotrophs. Why they are autotrophs? Auto means cell, trophism means to prepare. As plants prepare their own food, they are called as autotrophs. We are heterotrophs. These plants, then how these plants they prepare their own food? These plants prepare their own food by the process which is called as a photosynthesis. It is the only process during which O2 is released. Photosynthesis was carried in all green plants, some photosynthetic bacteria and Euglena. Euglena, it is a photosynthetic bacteria which was connecting link between plants and animal. When light is available, photosynthesis was done by Euglena, but light is not available. This Euglena it acts as a heterotroph, that is, it lives on other living organisms. Let us study about the ultrastructure of chloroplast. Chloroplasts are the green plastids which are present in all green parts of the plant. Chloroplast, it is the actually site where the mechanism of photosynthesis it was carried out. So chloroplast it is called as a structural and functional unit of photosynthesis. Chloroplasts, they are absent in prokaryotes. They are present in all green parts of the plant. They are in polymorphic shape. That means in some plants it may be cup-shaped, in some plants it may be reticulate, it may be star-shaped, or in Spirogyra it is in reticulate form. So let us study about the ultrastructure of chloroplast. Chloroplast it mainly protected by a double layer. That outermost covering it is called as a chloroplast envelope. As it is bilayered, outer layer it is called as outer membrane and inner layer it is called as a inner membrane. Both the membranes they are unit membrane. Then what is meant by unit membrane? Unit membrane means it is in lipoproteinous in nature. That means it consists of lipids and proteins. Both these membranes they have a space. That space it is called as a periplastidial space which was filled with a fluid which is called as a periplastidial fluid. Then the next part is the stroma. What is stroma? It is the colloidal semi-fluid matrix which is present inside the chloroplast. It is with 78 type of ribosome, RNA and proteins. It mainly contains the enzymes which are required for dark reaction. And the most important point is it mainly contains the DNA which is called as a plastidome. And what is plastidome? Plastidome is the circular naked closed DNA. It is the characteristic feature of chloroplast. Then the next part is the grana. This chloroplast, it mainly contains dark cylindrical stack of coin-like structure which are called as a grana. In each chloroplast, there are about 40 to 60 these stack of coin-like structures are present. The single, it is called as a granum. Each granum, it mainly contains about 2 to 10 these disc. Each disc, now it is called as a thylakoid. What is the function of grana? Grana, it is the actually site of light reaction. Then the next part is the intergranal lamellae. What is intergranal lamellae? These grana, they are interconnected with each other by fiber-like structure which is called as a intergranal lamellae. And then last part is the thylakoid. Each sac or each this disc, it is called as a thylakoid. What this thylakoid contains? This thylakoid mainly contains the contasomes. What is contasome? These are the circular or globular units which are present in the inner membrane of thylakoids, in which at the center there is a main pigment which is called. And last part is the contasome. What are contasomes? Contasomes, these are the small globular units which are present in the inner membrane of thylakoid. If we study the structure of contasomes, contasomes are actually, these are the globular units. Yes, these are the globular units in which there is a centrally placed chlorophyll A molecule. As we know, chlorophyll A molecule, it is called as a main pigment, or it is also called as a reaction center. This chlorophyll A molecule, it is surrounded by group of pigments. These group of pigments, they are called as a accessory pigments. What are accessory pigments? Accessory pigments are the pigments which only trap light energy, but they are unable to convert this energy into chemical energy. Yes, it is the structure of contasome. Where these contasomes are present? These contasomes, they are present in the thylakoid or inner membrane of thylakoid. This is a single thylakoid, second, third, fourth. Yes, this stack of coin-like structure, it is called as actually granum. This granum, it mainly consists of about 2 to 10 these globular or disc-like structures which are called as a thylakoid. In each chloroplast, about 40 to 60 this granum, singularly it is called as a granum, plurally it is called as a granum. In each chloroplast, about 40 to 60 grana are present. Thus, contasome, centrally placed chlorophyll A molecule, chlorophyll A pigment surrounded by accessory pigment. This contasome, it is present in the inner membrane of thylakoid. Thylakoid, it is the member of granum. Granum, member of grana, and all these grana, they are present in the matrix or in the stroma. Stroma, it is the colorful or it is the colorless liquid present in the chloroplast. Thus, we actually study the ultrastructure of chloroplast, in which chloroplast mainly shows outermost layer which is called as a chloroplast envelope, which is a unit membrane, means consists of lipoproteinous in nature, that is consists of lipids and proteins. It is smooth, it is selectively permeable membrane. Then the next part is the stroma. Yes, it is the site of dark reaction in which plastidome, 70S type of DNA, RNA and proteins are present. Then the next part, it's called as a grana. This grana, it is actually site of light reaction, and the stroma lamellae, that stroma lamellae which connects the granum, and at last the structure of contasome, where all the pigments which are necessary for mechanism of photosynthesis, they are present. Now we study the structure of chlorophyll. As we know, chlorophyll, it is the green pigment which is essential to do the process of photosynthesis. It is the structure of chlorophyll molecule. Chemically, chlorophyll A molecule is with head and tail. Head is with porphyrin ring which is with centrally placed magnesium. This head, it is hydrophilic in nature. While the next part is the tail. Tail, it is with long chain of hydrocarbons, and this tail, it is hydrophobic in nature. Now let us study some MCQs based on this content. The first MCQ is, which of the following is or are the raw material for process of photosynthesis? Yes, the options are H2O, CO2, light and chlorophyll. The correct answer is chlorophyll. Why? Because H2O, CO2 and light, they act as a raw material, but chlorophyll, it is the green pigment which is actually present in the plant. So it does not act as a raw material. Then the second question is, it is based on the chemical reaction of photosynthesis. Yes, what is the question? How many molecules of water are needed by the green plant to produce one molecule of hexose sugar? The options are 6 molecules, 12 molecules, 24 molecules and 1 molecule. Yes, we study the chemical reaction of photosynthesis, in which there is a chemical reaction between carbon dioxide and water. There actually needs 6 CO2 molecules and 12 molecules of water. So the correct answer is 12 molecules of water. They are needed for producing 1 molecule of hexose sugar. Then the third MCQ is, arrange the sequence from in to outwards with respect to chloroplast. Yes, we actually study the ultrastructure of chloroplast, in which we study the chloroplast envelope, then stroma, then grana, then thylakoid membrane, then contasome. Yes, we have to arrange these parts from inwards to outwards. The options are given below. Yes, the correct option is second option, that is a chlorophyll, contasomes, grana and chloroplast. Yes, we study that chlorophyll pigments, they are actually present in the contasomes. These contasomes, they are present in the thylakoid. Thylakoid, it is part of grana, and these grana, they are present in the chloroplast. Yes. Now, the next point is photosynthetic pigments. What are photosynthetic pigments? The pigments which are involved in the process of photosynthesis, they are called as the photosynthetic pigments. These photosynthetic pigments, they are complex colored compounds which has ability to absorb certain wavelength of light and reflect another wavelength of light. Where these photosynthetic pigments are present? These pigments, they are actually present in the contasomes. Where are these contasomes? These contasomes, they are present in the inner membrane of thylakoid. What is thylakoid? Thylakoid, it is the part of granum. Where is granum? Granum, it is part of grana. And where are these grana? These grana, they are present in the stroma, and stroma, it is part of chloroplast. Thus, the pigments which are involved in the photosynthesis, they are actually present in the contasomes. There are different types of photosynthetic pigments. As in our syllabus, main 3 types of pigments are described as, first type is the chlorophyll, the second one is carotenoids, and third one is the phycobilin pigments. Chlorophyll, the first pigment. These chlorophyll, they are green colored pigments which are mainly insoluble in water, and they are soluble in organic solvents like ether and acetone. Chlorophyll, it is again of there are about 7 types of chlorophyll, as chlorophyll A, B, C, D, E, bacteriochlorophyll and chlorobacterioviridin. Chlorophyll, they mainly reflect green light, so they appear greenish in color. Maximum absorption of chlorophyll, it is done in red color, which is followed by blue and violet color. Chlorophyll, it is again of, in our syllabus, there are 2 types of chlorophyll pigments are described as chlorophyll A and chlorophyll B. How we differentiate between chlorophyll A and chlorophyll B? As chlorophyll A, it is bluish green in color. Its empirical formula is C55H72O5N4 and Mg, and this chlorophyll A, as we know, it is called as an essential pigment. Why this pigment it is called as essential pigment? As chlorophyll A, it has ability to absorb light energy and convert this energy into chemical energy. While chlorophyll B, it is yellowish green in color, and its empirical formula is C55H70O6N4 and Mg. This chlorophyll B molecule, This chlorophyll B pigment, it is also called as a accessory pigment, and it is unable to convert light energy into chemical energy. This chlorophyll A pigment, it is mainly present in all photosynthetic organisms except some bacteria, while chlorophyll B pigment, it is present in all green parts of the plant except some blue-green algae and some bacteria. Then next main pigment is the carotenoid. This carotenoid, it acts as a accessory pigment and protects chlorophyll from photooxidation. Carotenoids, they are again of two types as carotene and xanthophyll. These carotenoids, they are orange-red in color. The main source of carotenoids is carot in the form of alpha, beta, gamma and theta carotene. Then phytotin, lycopene, which are mainly present in the ripe tomatoes. Carotene, it is observed in orange color. Its empirical formula is C40H56. This carotene, it is also called as an accessory pigment, and it is mainly present in the lower and higher plants. And the next pigment is the xanthophyll. Xanthophyll, it appears yellowish in color. Its empirical formula is C40H56 and O2. This xanthophyll, it is also called as a accessory pigment which is present in lower and higher plants. Then the third main pigment, it is called as a phycobilin pigments, which are mainly present only in the algae and algal members, that is members of Rhodophyta, Cyanophyta and Cryptophyta. Phycobilins, they are again of two types as phycoerythrin and phycocyanin. These phycobilin pigments, they are soluble in water. The phycoerythrin, it is red colored pigments. Its empirical formula is C34H46O8 and N4, which is seen in red algae, that is member of Rhodophyta. Then the last pigment is phycocyanin, which is blue colored pigment. Its empirical formula is C34H44O8 and N4, which is mainly present in cyanobacteria, that is members of Cyanophyta. Let us study the MCQs which are based on this content. The first MCQ is, which one of the following functions as a reaction center in photosynthesis? Yes. How we go towards the answer? As we know, what is the reaction center in photosynthesis? Yes, we study the different types of pigments: chlorophyll A, chlorophyll B, xanthophyll, carotene, phycobilins. Out of these pigments, chlorophyll A, it is called as a reaction center, and it is able to convert light energy into chemical energy. Then what is the empirical formula of the chlorophyll A molecule? The correct answer is the C. This is the empirical formula of carotene. This is the empirical formula of xanthophyll. This is the empirical formula of chlorophyll A, and this is the empirical formula of chlorophyll B. As the question, reaction center, we know chlorophyll A, it acts as a reaction center. So the correct answer is C55H72O5N4 and Mg. The next MCQ is, color that we see in leaves is due to presence of. Yes, we know plants are green in color due to presence of chlorophyll. But there is not chlorophyll is the only pigment present in the plant. Plants are green in color due to presence of main pigment and also in presence of due to presence of accessory pigment. The options are chlorophyll A, chlorophyll B, xanthophyll and carotenoids. Yes, the correct answer is the D option, that is first, second, third and fourth. That means in plants, all these pigments, they are present in high or low proportion, which gives green color to plants. Study the next point, that is nature of light or quality of light. Yes, as we know, CO2, H2O, chlorophyll and light, they act as a raw material for the process of photosynthesis. Then what is the source of light for the process of photosynthesis? Yes, sun or sunlight, it acts as a source of light for the process of photosynthesis. Mainly light, it travels as a stream of tiny particles which are called as a photons. Light energy, it mainly shows dual nature. That means it behaves as a waveform. by wave theory, or during interaction it acts as a stream. Sun mainly radiates electromagnetic radiations, which consist of two types of radiations as visible radiations and invisible radiations. Invisible radiations are the radiations from which mainly contains gamma rays, X-rays, ultraviolet rays, infrared rays and radio waves. These invisible radiations, they are harmful to life, and due to which photosynthesis does not occur during these invisible radiations of light. Then in which range the process of photosynthesis it was carried out? The process of photosynthesis it was done in visible radiations, that is the wavelength between 390 nm to 730 or 760 nm. In this region or in this range of wavelength, the process of photosynthesis it was carried out. So this region it was actually called as a PAR, that is a photosynthetically active region. Thus, the quality of light, yes, if we know about the quality of light, then light mainly shows 7 colors. Yes, we know it. Light mainly colors, or visible light, it mainly shows 7 colors, as famously it was known as the VIBGYOR, that is violet, indigo, blue, green, yellow, orange, followed by red. Thus, the photosynthetically active region in which actually the process of photosynthesis it was carried, it is the range between 390 nm to 730 nm. This slide shows mainly absorption spectrum, that is curves showing different wavelengths of light absorbed by the pigments. Yes, chlorophyll A molecule it absorbs wavelength of about 400 nm. Then chlorophyll B molecule it absorbs wavelength about 500. Then followed by carotenoids, which mainly absorb wavelength of about 680 to 700 nm wavelength of light. This slide it mainly showing the rate of photosynthesis of at different wavelengths of light. Yes, the rate of photosynthesis it was maximum in red light, followed by blue light, which is followed by violet light. Let us study. The next point is photoexcitation of chlorophyll A molecule. I repeatedly said that chlorophyll A molecule it acts as a reaction center, or out of these all pigments, chlorophyll A it is called as a main pigment. Why chlorophyll A molecule it is called as a main pigment? This chlorophyll A molecule it has the ability to trap light energy and also convert this light energy into chemical energy. But the remaining pigments are not converted this light energy into chemical energy. So chlorophyll A molecule or pigment it is called as a reaction center, or it is called as a main pigment. Then how the photo excitation of chlorophyll A molecule it was done? Initially chlorophyll A molecule it was in ground state, but when it absorbs light from the sun it becomes activated and it becomes energy rich. That state it is called as a excited stage. The first stage is the ground state. Ground state is the state in which chlorophyll A molecule it is in ground state. That means it has a not energy rich electron, but when it absorbs light energy from the sun it becomes excited, and that state it is called as a excited state. Now this chlorophyll A molecule when it becomes excited it expels energy rich electron, and this energy rich electron it was transferred to different transfers and it was accepted by during different acceptors. This electron it is not a normal electron. This electron it was energy rich electron. So this energy rich electron when it is transferred to different transfers, there is a synthesis of energy in the form of ATP, and this energy in the form of ATP it was used to carry out the process of photosynthesis. When chlorophyll A molecule loses that energy rich electron, this chlorophyll A molecule it again comes in the ionized state. Now this chlorophyll A molecule remains in ionized state only not more than 10 raised to minus 9 seconds. It again absorbs light energy from the cell and it again become in active form and expel energy-rich electron. Thus, light energy, it is converted into chemical energy for synthesis of ATP. Thus, at the end, yes, we study why chlorophyll A molecule, it is called as a reaction center, or how photo excitation of chlorophyll A molecule takes place, or why chlorophyll A molecule, it is called as a main pigment. Yes, we study that this chlorophyll A molecule, it is with, firstly it is in ground state, that is it is initially in ground state when there is a absence of light. When this chlorophyll A molecule, it absorbs light from sun, it become active, that means it is excited. When this chlorophyll A molecule, it become excited, it emits energy rich electron. When this chlorophyll A molecule expel energy rich electron, this chlorophyll A molecule, it again come into ionized state, or it again become positively charged. But this chlorophyll A molecule cannot remain in the ionized state for more than 10 raised to minus 9 seconds. Thus, the energy which was stored in the electron, it was transferred for synthesis of energy in the form of ATP, which was required to carry the process of photosynthesis. Let us study the main or important MCQ from this content. What is the wavelength of radiations in visible spectrum? Yes, we study the nature of light and we study the quality of light. Yes, the correct answer is the 390 to 760 nm wavelength. It is the visible radiations range wavelength during which the process of photosynthesis it was carried out. So this region, it is also called as a PAR, that is a photosynthetically active region. Let us study the next point, that is a photosystem and its types. Then what is photosystem? Photosystem, it is a group of main pigment with the accessory pigments. Here is the diagram which shows, here is the main pigment, that is a chlorophyll A, which is also called as the reaction center. This chlorophyll A molecule, it was surrounded by the accessory pigments. Thus, the group of main pigment with the accessory pigment, it is called as a photosystem. Photosystem, they are again of two types, as PS first and PS second. Where these photosystems are located? These photosystems, they are located in the thylakoid membrane of the chloroplast. As I told you, photosystem, they are of two types, PS first and PS second. PS first, it is made from light harvesting component and reaction center as a chlorophyll A. Here in PS second, also at the center there is a reaction center which is chlorophyll A and surrounded by accessory pigment. Then what is the difference between PS first and PS second? The main difference is in PS first, chlorophyll A molecule, that is the reaction center, it has a wavelength about 700 nm. While in PS second, the chlorophyll A molecule, it has a wavelength about 680 nm. Then how we define PS first and PS second? We define PS first as, PS first, it is the photosystem in which chlorophyll A molecule, which have a wavelength about 700 nm, it acts as a reaction center surrounded by accessory pigments. How we define PS second? PS second, it is defined as chlorophyll A molecule, which has a wavelength about 680 nm. It acts as a reaction center and it is surrounded by accessory pigments. Thus, there are two types of pigment systems as PS first and PS second. Photosystem, PS system as centrally placed chlorophyll A as a reaction center or main pigment surrounded by the accessory pigment. Only difference is that in PS first, chlorophyll A molecule or reaction center, that chlorophyll A molecule, it has a wavelength about 700 nm, while in PS second, this chlorophyll A molecule, it has a wavelength about 680 nm. Thus, there are two types of photosystems which are involved in the process of photosynthesis. The next point from this topic, photosynthesis, is the Hill's reaction or photolysis of water. What is photolysis of water? If we dissect the word photolysis, photo means light and lysis means breakdown. There is a breakdown of water molecule in presence of light. It is called as a photolysis of water. Yes. As we study the chemical reaction of photosynthesis. What is chemical reaction of photosynthesis? 6CO2 plus 12H2O. There is a formation of carbohydrates or food in the form of C6H12O6 plus 6H2O, and O2 is released as a gas. All this process takes place in presence of light and with the help of chlorophyll. Yes, it is the chemical reaction of photosynthesis. How we define photosynthesis? Photosynthesis, it is the process, or it is the anabolic process in which there is a reaction between carbon dioxide and water in presence of light. With the help of chlorophyll, food is synthesized and O2 is released. The question arises, yes, O2 is released. This O2, which is the source of this O2? That means this O2, it is released from CO2 or this is released from H2O. As O2, it is the content of both carbon dioxide and H2O. The question arises in the minds of scientists that the O2 released during photosynthesis, it comes from either CO2 or H2O. The answer is the Hill's reaction or Hill's experiment, and it was actually studied by the content or by the point that is called as a photolysis of water, or it is also called as a Hill's reaction. Yes. The old concept, according to the old concept, it was thought that, it was suggested that O2 released during photosynthesis, it comes from splitting of CO2 and not from H2O. According to old concept, the carbon from CO2, it is reacted with H2O to form carbohydrates, and there is no splitting of H2O. But Van Liel first suggested that O2 released during photosynthesis, it comes from splitting of H2O and not from CO2. For his experiment, he used the bacteria which are called as a green sulfur bacteria. Yes, 6CO2 plus 12H2S, C6H12O6 plus 6H2O plus 6S. Van Liel first studied the bacteria which are called as a green sulfur bacteria. These are the photosynthetic bacteria which mainly does the process of photosynthesis, but they used a H2S instead of H2O. These bacteria, they mainly used H2S instead of H2O. When they does the process of photosynthesis, there is a deposition of sulfur. That means there is a splitting of H2S and not splitting of CO2. Thus, first Van Liel suggested that O2 released during photosynthesis, it comes from H2O and not from CO2. But Robert Hill first experimentally proved that O2 released during photosynthesis, it comes from splitting of H2O and not from CO2. As he first experimentally proved it to the world, that reaction, it was famously known as the Robert Hill's reaction or Hill's reaction. Then which experiment, how he does his experiment for clearing this? He does an experiment. What he done? He first take a beaker. Yes, in this beaker he adds isolated chloroplast. That means he only add chloroplast. Yes, he isolated chloroplast and he add this chloroplast in a beaker containing water. Yes, this beaker contains water. Robert Hill adds chloroplast in this beaker. Yes, this medium, it was completely free from CO2. He makes that medium completely free from CO2. He adds some ferric salt into this medium and keep this apparatus in presence of light. What he observed? After some time, he observed that there is evolution of O2 bubbles from this medium. Yes, O2 is evolved from this medium. Then he concluded that there is a splitting of H2O and not of CO2, as this medium is completely free from CO2. He also observed that this ferric salt, it was reduced to ferrous salt. That means there is splitting of water and there not splitting of CO2. Thus, he experimentally first time proved to the world that O2 released during photosynthesis, it comes from splitting of H2O and not from CO2. Thus, Robert Hill experimentally proved that O2 evolved during photosynthesis, it comes from splitting of H2O and not from CO2. Then last confirmation, it was given by Rubin and Cumming. As Robert Hill suggested evolution of O2 from splitting of H2O, it was actually confirmed by the Rubin and Cumming. What Rubin and Cumming done? They mainly used chlorella and mixed it into the water, and he kept it in the presence of light. But what they have done, they have actually used a radioactive isotope, that is O18 instead of O16. Yes, they only replaced the radioactive isotope of O18 instead of O6. Rubin and Cumming done. In first reaction, we actually see the 6CO2 carbon dioxide, which have a radioactive isotope of oxygen, reacts with H2O, food is synthesized and O2 is released. But the O2 released, it has a not radioactive isotope. It has a normal isotope of oxygen. But in next reaction, we actually see that in H2O and water molecule, it is replaced by radioactive isotope O18. Yes, at the end of photosynthesis, we see that there is an evolution of O2, and the evolved O2 gas, it has a radioactive isotope, that is a O18. That means in the process of photosynthesis, there is a splitting of H2O and not of CO2. Thus, the world accepted that O2 released during photosynthesis, it comes from splitting of H2O and not from CO2. Thus, we actually revised that this point. What is Hill's reaction? Robert Hill experimentally first proved that O2 released during photosynthesis, it comes from splitting of H2O and not from CO2. As there is a breakdown of water molecule, and this breakdown of water molecule, it occurs only in presence of light, so it is called as a photolysis of water. According to old concept, it was thought that O2 evolved during photosynthesis, it comes from CO2 and not from H2O. But first, Van Niel, by using green sulfur bacteria, suggested that O2 evolved during photosynthesis, it comes from H2O and not from CO2. But Robert Hill first experimentally proved that O2 evolved during photosynthesis, it comes from splitting of H2O and not from CO2. He done experiment. What he have done? He take a beaker. In this beaker, he add chloroplast. Then he add water. Then he add ferric salt. And this medium, it was completely free from CO2. All this beaker, all the instrument, it was kept in presence of sunlight. What he observed? He observed that there is a evolution of oxygen bubbles. That means there is a splitting of H2O. Yes, This medium is completely free from CO2. There is an absence of CO2. There is no CO2. O2 is evolved. That means there is a splitting of H2O and not of CO2. Thus, he experimentally proved to this world. And his suggestion actually confirmed by the scientists, that is called as a Rubin and Kamen, where these Rubin and Kamen, they used a radioactive isotope of oxygen instead of normal oxygen. Thus, it was accepted that O2 evolved during photosynthesis. It comes from splitting of H2O and not from CO2.